Okay, so we're back, everyone, and what we want to talk about today is something called cross-presentation of antigen. So if you remember from last day, we talked about the two main pathways, and we have a class 1 pathway over here, which is mainly processing of antigens from within the cell that are manufactured within the cell, for example, in the cytosol. And we have the class 2 pathway over here on the right, when antigen is taken in via phagocytosis, for example, from outside the cell. So a way of dealing with two different kinds of pathogens, really. The intracellular viruses, for example, when they're actually making their proteins inside the cell, and for bacteria or other parasites that are taken in by sampling from the environment outside the cell. But sometimes it would be really convenient to be able to get some of these peptides from this exogenous pathway into the cytosolic pathway. And the reason for that is sometimes there are viral particles in fluid phase, the bloodstream, for example. And so it would be really nice if those could be loaded both on class 1 and class 2. Well, under the right situation, when the right stimuli are applied, this can happen. And it can happen mainly in dendritic cells. So that's what we see here. That's been well documented. Now, experiments in vitro have also shown that neutrophils, for example, can do cross-presentation, but that's not yet been seen in vivo. So the verdict's a little bit out on that one. But here's essentially what happens. The exogenous antigen is a viral particle, for example. This is going to be taken into the endosomes, as we've discussed before. But under certain situations, there are mechanisms that we don't yet fully understand that shuttle this set of peptides into the ER. So once they're in there, now they are free to load onto these MHC class 1. This is crossover pathway. And so what this means in practical terms is that if this is a viral infection, you're going to get both cytotoxic T cells activated and you're going to get the T helper cells activated. And that's ideal. That's what you want. Because remember, what happens is DCs are situated around the body. Let's think about the skin, for example. Lots of these dendritic cells, called Langerhans cells, that are in the skin, they are sampling antigen, so they may pick up their viral particles, for example. That dendritic cell is then going to migrate to lymphoid tissue, such as a draining lymph node or to the spleen, depending on how the virus is seen. And it's within the lymphoid organs that the interaction takes place between the different cells required to make an immune response. So you need a place where the interactions can be facilitated, where all the cell types necessary are held together in a framework, and that's lymphoid tissue. Once the appropriate activation has occurred, then the effector cells, let's talk about effector T cells, cytotoxic T cells, for example, they can migrate out of that draining node and they will go back to the site of infection to deal with the pathogen. They migrate there because certain signals, for example, inflammation at the site of infection, will draw them there using chemotactic factors, for example. So I say all that just to remind you where these reactions take place and that you do need all the cell types together. And so let's look at the next slide, because this kind of shows you what might occur. For example, this might be sort of a mini situation of what would happen in a draining lymph node. So we've got our DC here, our professional APC. We've got the exogenous antigen, and this exogenous antigen is going to come in through the cell via some kind of a receptor. So here, for example, it might be antibody. If this was a B cell, it might be some kind of a TLR. And normally speaking, this kind of an antigen is going to go through and activate T helper cells because it's loaded onto MHC class 2. And that T helper cell is going to produce certain sets of cytokines once it becomes active. Meanwhile, if some of these peptides, say viral peptides, can get shuttled via cross-presentation and get loaded onto class 1, that means the cytotoxic T cell can begin its activation as well. And that can occur without the antigen presenting cell, the dendritic cell, actually being infected by the virus itself. It's simply sampling the virus. If there wasn't this cross-presentation pathway, you would have the T helper cell activated, but you really want to have also cytotoxic T cells, but you wouldn't have a way to do that. So step 1 is kind of get it loaded onto the helper cell. This becomes activated. This helper cell can now give and provide the final stimulatory signals to fully mature this cytotoxic T cell so it can do its killing. This second step is called DC licensing by T helper cells. So essentially, the helper cell has given the cytotoxic cell a license to kill. And that can happen all within a framework like this if cross-presentation can happen. And if cross-presentation doesn't happen, then these two signals may also occur independently on other cells within a lymphoid organ. But it's really nice if you can have cross-presentation. That just facilitates that entire activity. All right, so hopefully that makes sense to you. Now, let's just spend a little bit of time, and we're going to talk about non-peptide antigens. So we've talked a lot about peptides, and there are obviously a lot of peptides that can be and need to be displayed to the immune system, both self and foreign. But what about things like carbohydrates and lipids? Well, it turns out that these also have a way of being displayed or shown to the immune system. So let's look at an example here. So here's a bacteria. And this bacteria has both the red, which would be the protein components, and it also has lipid components. Now, this might be something like a Mycobacteria tuberculosis, which has quite a thick outer wall, which is made of lipids and mycolic acids. And so these will be recognized by certain TLRs and taken into the endosome. And the lipids and the proteins will be broken down by the proteases that we talked about last time. The peptides are loaded onto MHC class 2. And now, as it turns out, these lipids are loaded onto another kind of molecule, and they're called CD1. But if you look at this, you can see the CD1 molecule looks a whole heck of a lot like MHC class 1. They're part of the same immunoglobulin supergene family. They have a binding pocket, and they also associate with beta-2 microglobulin. But the overall structure is unique. And so this is CD1. And you can see here CD1 displaying a lipid antigen. So now let's think about the different kind of T cells that can be activated. So we know MHC class 1 activates your cytotoxic cell. We know that MHC class 2 activates the various helper cells. And as it turns out, the CD1 activates other subsets of T cells, such as gamma delta T cells and NK T cells. So now you've got the complete ability to display all kinds of antigen on the surface. They will stimulate the appropriate kind of T cell responses or combination of responses, because sometimes, like this example, a peptide is going to be displayed and a lipid is going to be displayed. So you're going to get your gamma delta T cells, and you're going to also get your cytotoxic T cells. So you're going to help to optimize your immune response. And we can look a little bit in more detail at what the CD1 molecule looks like. So over here on the left, you can see its kind of general structure. You see the beta-2 microglobulin. And these A, C, and F, and T are showing you the tunnels or the deep binding clefts within this pocket. And this one here, T, is quite interesting because it's a very deep pocket or tunnel, and it allows highly hydrophobic residues of lipids or carbohydrates to fit into there, nice and snug into that pocket. Now, as I said, these look a lot like MHC class 1, but these are not polymorphic. They have very little polymorphism, but there are 5 human CD1 genes known, so you do get some diversity there. They function more like an MHC class 2 in some ways. So you can see here is an MHC class 2 displaying peptide. That's what you're used to looking at. But here you can see a CD1 and it's displaying lipid. And if you notice here, there is a very deep pocket where this highly hydrophobic residue can go down. So it's sitting in that sort of T tunnel that you see here. So again, different T cells are going to recognize these unique structures, either here of MHC or peptide, or over here of the lipid. Okay, so let's just summarize sort of this section before we move on to thinking more about the TcR and the various T cells. So T cells must have antigen peptides or lipid carbohydrates presented to them for activation. Understanding the molecules that perform this presentation is crucial to understanding the T cell activation, because then we can talk about the cytotoxic T cells, the helper cells, the gamma deltas, NKTs, and all of the different kinds of phenotypes of cells, and you're going to understand how they're displayed and how that activates various T cell subsets. Also understanding how intracellular and extracellular antigens, some will just be antigens, some will actually be pathogens, are processed for presentation, gives us insight into why different T cells are stimulated and activated during immune responses. So hopefully you've been able to capture that bit of information. Okay, so that's the end of the section, and we're going to move on here to the next bit in just a moment. Okay, so now we're moving on to the section entitled T cell receptors and signaling. There's 9 topics in this section. So we're going to look at the early evidence for the T cell receptor, the structure of this receptor, the complex that it forms and the accessory molecules, how it signals, the genes, the interaction with MHC. We'll talk also about MHC and TcR in terms of the role in graft rejection, T cell development, and finally T cell activation, differentiation, and memory. Let's get started. Remember to read the appropriate chapters. I think that's quite important, given that we have to be just doing videoed online. It's not quite as good as in person. So please read the chapters. I think you'll find that quite helpful. One of the things you're going to find, and you really need to take note of, is that B and T cell receptors have similar genetic architecture. And this architecture is completely different than what we talked about for MHC. MHC is polymorphism that's generated by Mendelian inheritance. B and T cells develop their diversity through VDJ joining of gene segments. So it's quite different. Make sure you've got that clear in your mind because it's quite important. So the BCR was discovered first, and that was relatively easy because when B cells become activated, they produce copious quantities of immunoglobulin. And so that's the receptor. You can get your hands on it. There's lots of it. Plasma cells can produce thousands and thousands of antibody molecules every second. But the TcR is not secreted. And so it was a little bit more difficult to take a look at it and figure out, what's the structure. But there were some experiments in the 1970s and 1980s that provided evidence that there was such a structure. And then the debate started as to whether it was sort of one receptor on the TcR that saw both antigen and MHC, or was it two? Were there two separate receptors for this? One receptor for antigen, another receptor for MHC. And so the debate raged on, but finally, this was resolved by experimentation. And that's why we get in the lab and we do the experiments. We look at the data. And so these experiments were done by two well-known immunologists, immunogeneticists actually, Kapler and Merrick. And they proved that indeed it's a single receptor. And we're going to look at the experiment actually that they did. I know sometimes you guys don't like necessarily to see all the experiments. You just want to know the bottom line. But I think it's really important that you learn how to think your way through how you might design these experiments, because for some of you it might be your job in the future to get into a lab and actually set up some of these experiments. So I think it's helpful if you know how it's done, and the best way is to provide you some really good examples. And so we're going to have a couple of examples of really good experiments that helped us better understand what was happening with the T cell. So what happened here is these researchers took two cells. They took a T cell, which is specific for KLH. That's an antigen that immunologists like to use. It stands for keyhole limpet hemocyanin. You may or may not remember that. And it came from an H2 mouse with the F MHC haplotype. So that meant that the cell is KLH plus IA, if we're talking about class 2 of F. These researchers also happen to have a hybridoma. Now, a hybridoma is an immortalized cell. In this case, it was a T cell hybridoma. Hybridomas are great because they never stop growing. You can keep them forever. You can do all kinds of experiments and not run out of cells. You don't have to keep going back to the mouse. So they're really handy. So they had a T cell hybridoma, but it came from an H2K mouse. And in this case, this was restricted, or the MHC framework that we talked about for this was a different antigen, OVA, which is ovalbumin. It's a chicken antigen, and its MHC restriction was IA of K. So we've got these two cells, and what these guys thought to do, which was pretty ingenious, simple. Sometimes these things are simple, but they're genius when you think about it. They fused them together. And they knew then there was going to be two possibilities. The cell, the fused cell, would either look like this cell on the left with two separate receptors, or it would look like the cell on the right with a single receptor for MHC and antigen. So if it's two receptors, when you fuse this, you'd actually have 2, 4, 6, 8 molecules on the surface, because you'd have to have a receptor for the MHC, for OVA, the ovalbumin. You'd have to have a receptor for the MHC part, IAFK. You'd have to have another receptor for KLH, and you'd have to have another receptor for the IA of F. And so I guess that's just a duplicate, kind of showing how they can be shifted around here. So this cell here is H2K and F. You can see the superscript F and K. So if there's two receptors, they call this the dual receptor model. But if there's just one receptor, you won't have these individual receptors around the outside of the cell. Instead, you really only have two kinds of receptors. You'll have a combined receptor that recognizes OVA plus IA of K, and you'll have another receptor, which came from this cell, that recognizes KLH plus IA of F. This is called the altered self model. So they said, okay, there's only two possibilities here. Let's do an experiment and see what we actually end up with. And that's what you're going to see down here on the bottom. So the various antigens are OVA and KLH. Then they add MHC on an antigen presenting cell. So they have antigen presenting cells that can activate these fused T cells. And the MHC on the antigen presenting cells, they had IA of K, IA of F, and this was both in association with the OVA and the same with KLH. So these are all the possible combinations that they had. So they did the experiments 1, 2, 3, 4 times. 4 different experiments. They repeated them a couple times to make sure it all works, but that's the model. And then they said, okay, if it's a dual receptor model, here's the response we would expect. If it's like this, then any combination of these will give a positive response. The fused T cell will proliferate and produce cytokines, for example. However, if it's an altered self, it won't. Only when the OVA antigen is seen in the context of an IA of K MHC will you get a response. And KLH is seen in the context of IA with the superscript F, as seen in the two original cells. But you won't get a positive response when OVA is presented here in this context of IA of F or KLH and IA of K, because that's not how it was in the original. So they performed these experiments and then they looked to see what the actual response was. And it was absolutely clear that they only saw a response from these fused T cells when it was as in the original form on the cells. As you see here, OVA in the context of IA of K, right? That's what that T cell receptor could see, or KLH in the context of IA superscript F. But it did not display these other combinations as you would if there was all kinds of separate receptors. So that settled the issue. And now they could go on and study these in a little bit more detail and learn about the structure. So They were then able to use antibody monoclonals and pull these molecules off the cell surface to study them. And we'll talk a little bit more about that. But first, let's just have a look and compare antibody or immunoglobulin, what you see here, with the T cell receptor, which you see here. So you can see just at a glance, they have some similarities. There's this conserved region, and that's also seen here, or constant region domains, they're often referred to. And this makes them part of the same family. You see that they have variable region domains here and here, but you don't have what's called the heavy and light chain. Instead, you have, in this example, an alpha and a beta chain. Sometimes this will be a gamma and a delta chain, but they're not really heavy and light chains per se. You can see these are joined by disulfide bonds. You can see that there's a transmembrane region and that this piece right here is positively charged, and that will become important in a minute. And then you have your cytoplasmic tails. Cytoplasmic tails are pretty short here as well. So, members of the immunoglobulin supergene family, they have variable and constant region domains, but not heavy and light chain domains. The variation in the amino acid sequence really occurs here in this upper area, because this is where you're going to bind the antigenic epitopes and MHC right there. Just as antibody binds to its native antigen up here using the variable portions, the same is true, but what it sees, the T cell receptor, is not native antigen, as we learned. It sees MHC plus peptides. It can be alpha beta, gamma delta. We have got some conserved carboxy terminal. This is important for cellular communication. We have the disulfide bonds helping to hold this whole structure together. And we notice that the TcR is monovalent. It's not here, we've got the bivalent, or if this would be IgM on the surface, for example, it could even be pentameric, but this is monovalent. We're going to see now that this TcR interacts with a CD3 complex via some of the positive signaling here, amino acids, in the transmembrane region. Okay, so here we see the structure. Let's move this up a little bit. There we go. So first of all, here is the TcR. We just had a look at that. But this is the CD3, and this is an essential co-stimulatory molecule. Just going to move my other screen here a little bit. There we go. Okay, so the CD3 is composed of a set of dimers, 3 dimers, and you can see that here. And these dimers are interesting because the first thing you'll notice is they have longer cytoplasmic tails. When you have a short cytoplasmic tail, really the molecules, it tells you right away that those molecules aren't great at signaling. And so this TcR is designed to engage the MHC antigen, but not to signal the cell to become fully activated unless you have a CD3 complex and unless proper signaling occurs through that. complex. And you'll notice that these little blue bars here, these are ITAMs. And you may have learned about these already in association with the B cell receptor. And these ITAMs refer to immunoreceptor tyrosine-based activation motifs. Because when these are activated, they can send signals, positive signals, through to the rest of the cell. And some of those signals are fairly complex. Oh, just a second, I'm going to pause that. My phone is ringing. That's one of the problems with this. Okay, so I apologize for that interruption, such as the troubles with online learning. Okay, so I just had mentioned these ITAMs, and these are activation motifs, but there are also things called ITIMs, and we're going to see that coming up here in a little while. And that is the inhibition motif. So it's immunoreceptor tyrosine-based inhibition motif. And these are how cells can be up and down regulated by kinases that can activate or inhibit what's going on in the cell. Now, just a little bit more about this complex. So first of all, it's essential for signaling. Signaling can't happen without this CD3 complex. And you can see that there are essentially a couple of different chains involved. So there's this gamma epsilon, you gotta look, make sure I can see that, epsilon delta combination. And then over here you see the zeta-zeta chain, so that's a zeta. And these have 3 of these tails, so that's super important in the signaling. And then you can see how these are actually arranged on the cell surface, for example, because it wouldn't just be on a linear form like this. These cells, the surface is very fluid. Molecules actually can move around on the surface. Molecules come up, molecules disappear, depending on what's happening. And they don't sit in a linear fashion in the lipid membrane. They actually can move around and get themselves situated so that you get this ideal sort of 3-dimensional conformation that can have a good look at what's going on in terms of MHC and antigen. So this picture up here on the right just kind of shows you how these may be interacting on the cell surface and forming more of a 3-dimensional structure. All right, so now look at this. This is a messy one, isn't it? Just kind of move that over there. Now, when you look at this, I'm sure you go, good grief. And the reason I put this up here is so that you actually get a feel for the complexity of what happens downstream after signaling. You know, we talk a lot about what happens up here on the surface of the cell. But for what's happening up on the surface of the cell, a lot more is happening down below. And it's complex and tightly regulated. And when you think about it, it works quite well because most of the time the immune system protects us very well and it doesn't make too many errors, all right? So this particular TcR, if we look at that, this is also going to have not only TcR and CD3, but it will have a CD4 in this case, or it could be CD8. Here's the membrane. Here are all of these tails. We're going to just kind of look, the ITAM sitting on them, this now in yellow. We're just going to have a little bit of a brief look. And you're not going to be expected to know this in detail, but just kind of to get an idea of it. So there are some important tyrosine kinases involved in activation of T cells. And two of the main ones that I think you should know are things like LIC and ZAP70. And you can see them here. So here's LIC right here. Here's ZAP70. So when this complex is engaged, you start to activate these molecules, and they actually can phosphorylate like so, these ITAMs. And when that happens, you actually change the structure of what's going on down here. So it's a whole series of these tyrosine kinases. You get other molecules, phosphatidylinositol, for example, protein lipase C, a whole lot. Calcium flux changes within the cells. And the end result of this is conformational changes. The molecules, all of these molecules begin to interact in the plasma membrane, and transcription factors are produced. The transcription factors, again, are diverse. Some are like NF-kappa B is a very well-known one. NFAT, another one. MAP kinase cascades can be activated. The transcription factors, which normally can't, for example, get into the nucleus, now they're activated in such a way that their structure has been modified, and they can actually get into the nucleus, and they can interact with appropriate pieces of the gene, and then you get activation of the cell itself. And so that's what allows the cell to regulate its survival, its proliferation, as well as its effector function. And so some of the genes that might get transcribed in a T cell would be IL-2 and the IL-2 receptor, for example. And this is key. IL-2 and the IL-2 receptor are critical for the survival of T cells so that they can go on and divide and become those effector cells. So that's just a brief look at this. I'm going to try and attach a small little video about the cell signaling to the course outline there. I'll put it in course link is what I'm trying to say. Okay, so a summary here is signal transduction is complex, but and I guess, but it's essential, but it happens. We won't delve into it too much here. We'll just have that brief overview that I gave you. Now, when you're thinking about B and T cells, I think the trick is to think about some of their similarities and think about their differences. And that will help you, I think, keep the two straight and why we have both B and T cells. So they use similar strategies once processes are underway. So a lot of the internal machinery is the same. Some of the basic structures are the same inside, outside even. They're a family of molecules, but the B cell sees the native antigen, the T cell sees the processed antigen. And that's so that we can deal with extracellular pathogens whole in native conformation. That's what the B cell does very efficiently. But we know not all pathogens are extracellular. Some are intracellular. Some are facultative intracellular. And so there has got to be another system, i.e. the T cell, that's designed to deal with intracellular, whatever it is, peptides, lipids, organisms that have these components. And so that's the job of the T cell, to deal with cells that become infected. Okay, so now I think we're going to move on. Let's see if we can get started. Let's just check the time here, how we're doing. I think we've got time to start this next section. And this is actually the genes. So just before we get started into that, we'll have another reminder of what the structure looks like. This is the protein itself, and we had the protein first, and the protein helps us to understand what the genes may look like in some ways. But as always, there's some surprises that come up with these particular genes. Because of the huge amount of diversity, we get some surprises. The geneticists really were intrigued by MHC molecules because how they generated diversity, and as well by B and TcR genes because of their unique mechanism as well of generating diversity. And that's actually how the field of immunogenetics was born, by people who wanted to understand genetic regulation of the immune system. And that's kind of one of my specialties. I actually did my PhD on MHC molecules and looking at how they control disease resistance in livestock. Anyways, that's kind of another story. Anyways, so immunologists were able to generate monoclonal antibodies that bound to specific parts of the T cell receptor, for example, parts you see here. And some of these monoclonals that were most useful were the ones that bound to conserved regions, because that meant you could pull down a whole lot of TcR. You weren't just pulling certain TcRs because this is much more variable. You were capturing a whole lot. And then they could be purified. Once they had them in their hand, you could purify them. Now we're going to have a look at this experiment, because that's another one of these experiments that's super important to understand. This is how the gene structure was determined. These experiments were done in 1984 by Hendrick and Davis. There was also another group at the University of Toronto that did similar experiments, and they used a technique, which you should understand, called subtractive hybridization. Again, these researchers took two different kinds of cells to start. They took a T helper cell clone and they took a B cell clone, knowing that these cells are related, they're similar, they're both lymphocytes, so they should have some genes in common. But they also knew they each had a unique receptor. By that time, they were aware the B cell had the immunoglobulin receptor, and they were trying to figure out what the genes look like for the T cell receptor, and they already had the structure basically in their hand because they had gotten that by monoclonals. So they took messenger RNA from each of these cells and they hybridized them together. Well, I should step back one step. Before they did that, from the T cell, they really knew that this was a membrane-bound receptor. So they just wanted to look at the messenger RNA that was membrane-bound polysomes. So that's about 3%. And so 97% mRNA is free in the cytoplasmic ribosomes, so they discarded that. And they just kept the membrane-bound piece. Then they said, let's fuse this mRNA and this mRNA to see what's in common and what's different. In order to do that, they first had to make a cDNA copy of this membrane-bound messenger RNA. They did that by a process called reverse transcriptase. And while they were at it, they gave it a radioactive label with P32. So now you've got P32 labeled cDNA, representative of the messenger RNA that's membrane-bound, going to encode for membrane-bound molecules from the T cell. They hybridize them together. In other words, let the pieces that are in common, that have homology, bind and get rid of that. So they used what's called a hydroxyapatite column to get rid of all of this hybrids, because they knew that this was the material that was common to B cells and T cells, and what they wanted was the unique T cell receptor. So they knew that the information, the genetic information about the T cell had to be in these remaining cDNA fragments. These would represent material that was unique to the T cell, and some of that had to represent the T cell receptor. This part here, where you do the hybridization step and you separate out these two types of RNA, this is the subtractive piece. That's the subtractive hybridization right here. So there's the hybridization, you let them join, and then you subtract. In other words, you take what's unique and separate it out from what's in common. You can do this with any kinds of cells, by the way. This just was ideal because of both being lymphocytes here. They were ended up with 10 different cDNA clones, one of which, or more, some of which, let's put it that way, were going to represent the genetic material, and so of the T cell receptor. And so then they went to the next part of their experiment, which was to use these probes. These are now referred to as probes, these clones, we call them probes, and they're going to be used in a process called southern blotting. And it's called southern blotting just because the man who discovered it, his last name was Sutherland. So southern blots. And what they decided to do is use these probes on genomic DNA that originated from various sources, either liver cells or B cell lymphomas, because they knew this should not have T cell receptors. So this was going to be like a negative control. And then they took genomic DNA from various different T cell clones that they had in stock. And they called these, in this case, A through F. So each of these clones should have a unique T cell receptor and therefore unique TcR genes. So let's look at the bottom panel first. So they chose a clone. They're just systematically using each of these clones. When they looked at some clones, clone 2, for example, they didn't see any difference here between the controls, the liver, the B cell lymphoma, with these various other T cell clones. They knew that wasn't what they were looking for. Oops. Okay, that's the end of the lecture time, but we're going to just wrap this up because we've got a couple of minutes here to spare. So they were looking for TcR which would be unique between the clones. And when they finally got to this cDNA probe here, so probe with cDNA clone 1, they saw the pattern they were looking for, which was that the controls were the same, but each of these T cell clones had a unique pattern that must be associated with a unique T cell receptor. And indeed, that turned out to be the case. And they could see the unique fragments. And I think the first chain that they were able to derive was the beta chain. So remember, TcR is alpha beta or gamma delta. And so that's the experiments that they did. Again, this is a fairly elaborate experiment, a lot of different techniques, but very well thought through. So I want you to be able to understand how they did that and why they did that. And the sort of the two main techniques, the subtractive hybridization and the southern blot working together. So the bottom line is genes are not rearranged on these controls, certainly not TcR genes were not rearranged, but here they could see that the genes were definitely rearranged because of these different patterns, each representing different VDJ region joining, how that would be different in each clone when you look at the genomic DNA and probe it with a piece of the messenger RNA. Okay, so hopefully that makes sense. And I think that's probably a good place to stop for today. Bye for now.